It's my pleasure to introduce the speaker for today. For this, we can see here the information and graph theoretical machinery for neuroengineering, and it's the Professor Behnaam Aazhang from Rice University, and we're looking forward to your talk. I can also say a few things about the Professor Aazhang. He's a fellow of the IEEE and the IEEES. He's a distinguished lecturer of the IEEE Communication Society. He has a PhD degree in electrical engineering and from the University of Illinois Urbana-Champaign. And in August 1985, he joined the faculty at Rice University, where he's now the Abercrombie Professor in the Department of Electrical Engineering and Computer Engineering. I don't think I want to say more about all the things you have achieved, so I'll just hand over the floor to you.
Thank you. Good morning, everybody. Is the microphone working? Excellent. Good morning. This is sort of the second installment of a two-part talk. Yesterday, I talked about a particular disorder, and then talked briefly about a couple of different methodologies to understand that particular disorder. I'll try not to make any reference to yesterday's talk, because I'm sure there are some people here that were not there yesterday, and they don't care about references to the talk yesterday. Therefore, I try to make this talk to be standing on its own. And with that said, I'm an engineer that is interested in neuroscience and interested in engineering problem has to do with our neurological system. And as an engineer, you need to work with neurologists and neurosurgeons and neuroscientists to be able to sort of focus on relevant problems. And these are the list of my collaborators that are in the medical center across the street from Rice. And today I'm going to talk about the work that we're doing with one particular neurosurgeon. And in particular, I'm going to talk about the work of one of my PhD students. And her picture is right there. It's hard to see in this photo, but she's there. And the work that I'm presenting today is basically very much new. We recently completed the work. Therefore, there are a lot of open questions. There are a few answers, but a lot of questions. And we are sort of done with this particular submission. And then if you're interested in having a copy of that submission, if you're interested to look at it further, I'm happy to share a preprint with you. And stepping back for a second, the age-old question is that how is our brain, how does it function? How does it work? And how is it able to do some fine motor control? How is it able to learn? How is it able to memorize, to recall, and many of these functionalities that our brain does every day? It's a mystery how we do it. And I'm going to sort of talk about this in the context of one disorder that sort of challenges a few of these functionalities of our brain. And to sort of check back to see what we know so far is sort of an interesting viewpoint. What we know is that our brain is made out of billions of neurons, and these neurons are cells, very similar to every other cell that our body has. And they function as an electrochemical entity, and they communicate with each other through signals that are electrochemical signals. And basically, it turns out that they are all very similar. It's basically a homogeneous system, homogeneous circuit. And but they do many different functions, and the functionality emerges from their individual action and how they communicate with each other, how they create the circuit. Therefore, it's not like they are set to do a particular thing. They do very similar things. There are many, many, many of them. Therefore, the functionality sits in their circuit, in their global activity. This is called emergence. It is a field of study by itself, how many simple things could emerge to do one thing that is very, very complicated. And you hear about this thing in bees and birds and many other natural organs. All right? And brain is a very fundamental example of an emergence. Now, it is made out of, as I said, millions of simple neurons, and then they work like a circuit. And as we can imagine, this is a circuit, therefore we can sort of try to understand how the circuit, how the elements of the circuit work. We can probe it. We can sort of put electrodes and record from these neurons and from the connection between these neurons, and then try to sort of understand the functionality from probing individual neurons or a population of neurons. Now, these neurons, for a human brain, we have a lot of neurons in the billions. And as I said before, the functionality comes from the interactions. The interactions is at a very, very low speed, about in the hertz level, not in gigahertz or higher speeds. And it's all about the connection, the synapses between neurons. There are about 100 trillion synapses in our brain, which is an incredibly large number. And they're trying to understand and decipher the activity, the functionality from this millions and billions of neurons is a very challenging problem. One of the challenging issues is that we don't have access to that. We cannot just get inside the brain easily and try to understand functionalities and all that. And that's obviously one of the big challenges. Now, people have thought about the connectivity of the brain, the circuit and all that, and then they have identified a few different types of connectivity in the brain. Obviously, the first one that you could think of is just the actual connection, actual physical synapses between neurons. It's called anatomic connectivity. But then a lot of anatomic connections are not functional. There is another concept of functional connectivity, that a small percentage of them actually are connecting to each other to create a function. And then obviously the last one is what we call effective connectivity, which is basically the causal connectivity. Right? And these are the three different connectivity that people study in this particular domain. And there are a lot of challenges in this space. We have been interested in how our brain works for many, many, many years. And the grand challenges today, again, has to do with how we function, how the circuits behave and change as we learn and memorize and all that. And when there is when we are in a healthy state, we function a particular way, and then when we are in the disease state, we don't function the same way, and we function some other way, that we're trying to understand the differences between healthy and disease. It turns out that there are a number of different disorders, neurological disorders. Believe it or not, there are over 200 named and identified neurological disorders in existence. Over 200. I was shocked when I actually sat down and counted them, and it is amazing, and they're all very different from each other, and they're all affect people in a very, very different way. In our group at Rice, there are several of us that are focused on neuroengineering, and we have focused on a number of different disorders, trying to understand how the brain works, trying to understand how we have diseases and how we can sort of mitigate a particular disorder and all that. And as I said before, yesterday I talked about epilepsy, and today I'm going to talk about another disorder called aphasia, which is kind of interesting because it is obviously not as important as epilepsy, but it kind of challenges us in a very different way compared to epilepsy. It's called aphasia, and that's what I'm going to talk about today. Now, what is aphasia? I'm not sure if many people know what aphasia is. I didn't, certainly, till I got involved in this. And aphasia is basically a language disorder. That basically, as it says here, this is a line right out of Wikipedia, which is where information is these days. It's basically an impairment of our language system, that we cannot produce language. Very simple, but very debilitating for many, many patients. And obviously it's not very common, but it is certainly a disorder that affects many, many millions of people. And it often is because of injury. It often is because of stroke, that a particular region of the brain is not functioning anymore, and then the patients have trouble articulating, trouble producing language, right? And it is very understudied, right? The reason is very obvious. Why is it understudied? Is because there are, as you know, we cannot model our brain. It is just impossible to do because there's so many elements and all that. It is also hard to study because there are no animal models, right? You can imagine that this is a very unique functionality of humans, that we can speak, and we can produce language, and other animals can't. Therefore, if they could, we could have sacrificed rats or rodents or monkeys and learn from their language system. And unfortunately, they don't. Therefore, we only can learn from humans, which would be a very difficult challenge to really probe the functionality of our brain. Therefore, it is sort of a benefit of being human that we can talk and we can produce language. And that brings challenges. What we don't know and we would like to know is that how we process visual information, that when I see Mikhail and I say, oh, that's him, right? I mean, I saw him and I can produce his name, for instance, right? How we can process visual information, how can we recall the name, how can we articulate the word, and how can we sort of remember, articulate, and then give command to our muscles to produce his first name. Right? These are all interesting questions that there is very little knowledge about how that whole process works. Right? And that's what we're interested in doing. Right? Now, again, I'm very open to questions. If you have any questions, please stop me anytime in the talk. And I really much appreciate it if the talk was interactive and that I would not be the only one talking. Yes? Example.
Does it affect hearing impaired people who use sign language?
I don't think so. That would be a different issue. Okay. The question was, does this, I'm getting there, does this affect people who are auditory impaired? I don't think so. I think this is just a very sort of specific disorder that is seen with people who had a stroke and a particular region was affected. This is very different from stuttering and other issues that exist. You know, people who stutter, they see, they know what to say, but it doesn't work, right? This is very different. This is a very different disorder, maybe correlated, related with the stuttering or other impairments like that, but we're just focusing on this particular one. It's speech production, exactly. Which is, again, this could be similar to stuttering, right? But that's not what we are studying. Very good question. All right. Let's keep going, and then please stop me if you have any questions. We have 45 minutes. I've never given this talk before. As I said, the result is very new. Therefore, I don't know how long it's going to take to talk. And but we will stop sharply at 11. All right? And then we can talk later and all that. Now, let's step back for 1 second and see where we are. What do we know about language production? What people have done is that they have done fMRI and other CT scan and other technologies to try to understand how, maybe even EEG, how people sort of produce language. And what they have done is that they've studied sort of look at different regions and measure the power emanating from those regions, saying, okay, if there is power emanating from that region, that means that that must be an active region. And then they have measured that in high gamma region, over 60 hertz region, and they have a sort of a understanding of how our language production roughly works. And this is basically roughly how it works, all right? Ignore these dots for now, and just focus on this nice looking brain. And these different regions with different coloring scheme are different regions of the brain that have particular names and particular associations. And the point is that when we are basically looking at a particular object, our visual cortex, this area right here, is activated. They see some power in visual cortex when we see an object, right? And then when we are processing and trying to find out what this is, but basically the semantic region has been identified to be this region down the stream from visual cortex. Right? These are what we know so far today. And then this power transition continues and goes to another region, which is known as the Broca region, which is basically where supposedly speech is produced. All right? This has been known for a few decades by now. And then the last piece would be the actual motor cortex on the surface of the brain, closer to the surface of the brain, where the Broca region commands the motor cortex to basically control different muscles of our vocal system to produce the word Mikhail. All right? And that's the motor cortex, the last piece of this puzzle. And it turns out that after that, we go back to the auditory cortex. Basically, when we say the name, we can listen to what we just said. All right? And that would be a region following this one that actually produces the sound. All right? This is what we know so far. And the underlying mechanism of this picture is not known at all. How these neurons behave, what's going on, how information is processed, and all that. But it's only known at a very coarse level and very coarse study of this particular system. All right? Now, what we are interested in is just to follow up that sort of knowledge base to see what is going on underneath that and what can we say about that and how can signal processing, information theory, and graph theory help us understand what's going on and sort of at a deeper level, right? And to refine the earlier question, it would be to say, what are the underlying mechanism of a language system? What are the causal relationship between different regions? Is this true that this region produces some signals and the other region follows that up and continues it further? Is that truly the order? And the coupling of these different regions, we are also interested in that. And also we want to know how the underlying circuit changes, but the dynamics on the underlying system is also important to us. These are the kind of questions we want to answer for these kind of problems. All right? And we go back to the problem that this is only can be seen in human, and it's very hard to sort of have access to specific activity of human brain, basically. All right. Now, our colleagues basically put together this experiment, which is very intuitive experiment on human subjects, and trying to understand all of these questions that we talked about before. All right. Let's just go through this particular exercise. The subject is sitting, a person, and it is seen a picture of an alligator, depending on what continent you are, either alligator or crocodile. And this is the stimulus that is stimulated by this particular picture. And then the subject is told to say what you see on the screen, on the screen, right? Therefore, this is the stimulus onset right here. And then at this point, the patient knows that he or she has to articulate the word alligator, right? And that's that point. And then the patient is asked to focus on some dot on the screen, rest a little, and then a second image is produced, stimulated, and then is asked to just repeat the same thing, right? And this is the experiment. Let's say a patient goes through this thing and goes through hundreds of trials of this particular experiment. And we wanted to see how does this map to a particular functionality of different regions of the brain. Right? Again, you could do this thing while the patient going through fMRI or other modalities of recording, or EEG, basically a cap on their head to record the activities. But as you can imagine, all of those are coarse recordings, and they're very high noise recordings. Therefore, signal-to-noise ratio is very poor. And we have been fortunate to be able to have access to recordings underneath the dura, underneath the skull, which is much closer to the source of activity, and we get signals at a very high fidelity. All right? Now, this is kind of a lucky situation for us because the same surgeon that works on epileptic patients, as we discussed before, those patients are in the hospital for 7 days, waiting to have a seizure, and they have a lot of free time in the hospital in between seizures. And they volunteer to go through a language test. And then the recording happens while they're just waiting for a seizure to happen. And this is basically the story. The story is that these patients are in the hospital. They either have Utah probe type electrodes implanted, or they would have what we call ECOG electrodes placed on the surface of their brain, on the actual tissues, and they record about 200, 250 location of the brain, and then we have those recordings for hours, and sometime during the day, the patient goes through this test that I described a second ago. All right? Believe it or not, these are nice people that they love science, and they go through this exercise, and they volunteer to do this thing. And you can imagine that doing this thing for 500 times a day, it is very taxing on them, especially they're not necessarily happy to be there, but they do it anyway, right? And believe it or not, many of these patients have been exposed to other tests that are actually painful. There's one test that they do where patient is given electric shock, and they anticipate electric shock. Therefore, they see something and they get electric shock, and then they show them the same thing again. They're anticipating electric shock, but electric shock doesn't happen, and they're trying to find out how humans associate this picture with electric shock. Now, I've seen that particular experiment is painful for the patients, but they do it anyway because they love science. This is not painful. It's just that it takes a long time. It's kind of boring, right? But it's not painful, trust me. And all of these patients are epileptic patients because we don't have access to other patients. And we also don't have access to aphasia patients because they don't necessarily also have epilepsy. Now, the point is that for now, we want to find out how we produce language. And then once we completely learn how we produce language, maybe we could help aphasia patients by stimulating different regions of the brain or bypassing a region of the brain. And sort of if a region is affected by stroke, maybe we can bypass that and then carry the information from one region to another region and help the patients be able to produce language. But for now, we don't understand enough to be able to do that, and these kind of experiments are there to help us understand. All right, any questions? All right, let's move. This is the kind of signal. This is exactly the kind of signal that we get from different patients, and very typical. I am only showing, in this case, 5 channels. As you see here, there could be 250 channels. This is only showing 5 of them. And as I said earlier, there were 7 patients that have gone through this. Therefore, we have data on 7 patients. And some of the results I'm going to show have been averaged over 7 patients. Some results are for specific patients. But as you could imagine, in this kind of picture, we may have 250 time series that show something like this. And then we would have, let's say, 200 to 300 trials for each patient for a given day. Therefore, the combination of these two could give us the statistical, could help us with the statistical significance of our results. Basically, that we treat these trials as sort of, a particular realization of our time series, basically. And then worry about the fact that this is not stationary and all the other issues that we can discuss. All right? Once again, this is basically a spatial-temporal analysis that we have data from these experiments that goes across time as they go through production of the language. And we have data across different electrodes, across different regions of the brain. Therefore, it's a spatial-temporal time series that helps us analyze analyze our language production system. All right, now, again, this is the data that we have, and there's a lot of it for a long time, and you're interested in finding a way to sort of, in the best light, depict the data. Put the data in a framework that you can understand. Our framework is a graphical tool. Basically, put this data on graphs, right? And basically, graphical, tools or information theory tools or other tools like that, they're not magical. They don't create information. All they do is that represent the information in a way that you could understand better. You could infer different things, different quantities that you want to infer. They don't do magic. They just help you sort of package your information in a more efficient way. Now, what we have done is that we've looked at this problem, all these time series and all that, and then we created a graphical model for this. And our graphs have vertices and edges. The vertices are basically the electrodes again. The electrodes are the vertices, basically the time series recording of different locations. And edges, we have a lot of freedom as to what the edges could be, right? And there are two things that we have looked at so far, and there are other things that we could look at, but for now, I'm reporting on two things that we looked at. One is basically the coupling between the two time series at different frequencies. As you know, in neuroscience, people are very focused on different frequencies and different frequency bands, and what these different frequency bands could tell you about the data, about your experiment. And one favorite in this application is high gamma, higher than 60 hertz, tells you a lot about the underlying process. And we are interested in understanding the coupling at these high gammas, the coupling of the signals in this high gamma. And understanding the power of high gamma is already quite well understood. I'm not going to talk about that. But the coherence between time series at high gamma is a particular element that we're interested in. Another one is, again, causality. Is again causality because you want to know how the information goes from one region to another region, how one affects the other one, how one time series wakes up another time series, and then how is this analyzed. Therefore, these are potentially our two possible edges for our graph. Our graph could have possible stages. As you can imagine, coupling is bidirectional, but information flow is one directional, is possibly directional, right? Therefore, you end up with undirected graphs, and if you change the edges, we may end up with directional graphs, right? And these are two examples. It's hard to see in this picture that the bottom one is a directional graph, and the top one is a undirected graph, all right? But the arrows is hard to see in this particular depiction, all right? Now, more importantly than these graphs, we are interested in the dynamics of these graphs, how they change as we produce language, okay? Now, I talked about all of this before, and let's talk about these metrics that we are going to use today, both coupling and directed information. And these are some things that the tools that we have, and I'm going to talk about them a little bit in detail today. Okay? Now, the undirected one are basically coherency between time series. Coherency meaning that how are they synchronized and how are they related to each other. Right? And then this coherency you can examine it at different frequencies. All right? Let's just talk about that for a second. Now, remember that coherency as a concept is a signal processing concept, and it's been around for a long, long time. And it's basically, for linear systems, it's been defined this way, as the ratio of the cross power expected density of two time series. We have a time series X and time series Y, like our recordings, and the cross power expected density at a given frequency divided by the individual marginal power expected density of each of those two time series, right, at F. And you can show by things like Cauchy-Schwarz inequality that this quantity for every frequency is between 0 and 1, normalized. And just for a second, let's see what it means and what are some intuitions that we can have from that. If you think of your data, your x, your time series x and time series y, if they're linearly dependent on each other without noise, therefore they could follow an equation like this. The y would be some convolution of some linear system, a linear impulse response with x, right? That is an input-output relationship that is linear, right? If that's the case, the question is that, then what is the coherence between x and y as two time series, right? And that's the definition, right? If you bring out some basic linear systems, you remember that the cross power expected density between two time series at a frequency f, if they're linearly related, it would be defined as the transfer function of the filter h right here, the transfer function of that filter, times the power expected density of the input, right? Therefore, the cross is h times the power expected density of the input, right? Basic random process. And that helps us with this numerator. The denominator is the power expected density of the output times the power expected density of the input. Power expected density of the output is the transfer function of the filter in absolute value terms squared times the power expected density of the input, right? That's something that I teach in my graduate random process class, right? Easy. Now, if you plug in all of these things into this, what do you get? One, right there. Where is it? Oh, right there. You get one. This is beautiful. That means that if you have this linear system that has no noise, then the coherence between process x and process 1 is 1. The coherence is 1. That means that the linear system doesn't create new frequencies, and these two signals are coherent at the frequency, and they have a very nice, simple relationship between them, and very well understood. Is 1. Now, if your system has noise, or if system is nonlinear, you could imagine that in that case, it would not be 1. It would be something less than 1. And 1 minus this coherence could be telling you how much noise you have or how nonlinear your system is if you were able to figure this thing out for nonlinear systems. Kind of cute. It's a cute little result that is not ours. It is Behringer's result. It's kind of neat. It's kind of neat to revisit because I believe is underappreciated in our community. But anyway, I'm going to get off my soapbox here. And let's see an example. An example would be interesting to see if an example is not linear, right? Therefore, we have two signals, x and y, and they're related this way. Z is just noise, so some simple noise, and y is equal to x squared plus noise. In this case, because of the fact that these two are not linearly related, this equation is not going to help us. It turns out that we failed in coming up with a decent number, because we know that these things are somehow coherent with each other because of the definition, but this thing doesn't help us get that. All right? And the question is, how can we sort of think about a metric that would measure coherency among these kind of signals? Because we know that our brain is not linear, therefore we need to extend the definition of coherency for nonlinear systems. Right? And again, this is not our work. This has been around for a while. Right? Now, what people have done is that they say, OK, that's fine. Let's just think about mutual information as a quantity here. And basically, for years, people have time series, and they look at the mutual information between these two time series. And it works beautifully. It tells you all about the not coherence, all about correlation between the signals. And it works in a linear case, nonlinear case, Gaussian, not Gaussian, all that. Mutual information between two time series is defined right here. And this definition is basically a definition for time series and doesn't give you anything about frequencies. Doesn't tell you anything about frequencies, but it is just for time series. And but the reality is that in many disciplines, we are very interested in frequencies. In music, it's all about frequencies. In neuroscience, also people are very interested in different sort of analysis of neurological data at different frequency bands. right? And then mutual information is not helping us in that domain. What people have done is, okay, let's take the time series and take it to the frequency domain, and then look at the mutual information in the frequency domain. Because whether the index is time or frequency or space doesn't really matter to information, mutual information. It just gives you a number. It gives you a number based on statistics, no matter what the index is. Basically, this is your time series, and you can take it to the Fourier domain with this kind of a fancy integration, fancy Fourier transformation, and then you can have now the data in the frequency domain. All right? And let's see what we get. And then for these, series, which are like now not time series, they're frequency series, then you can look at the mutual information between them if you have the densities and all that. All right? Now, it turns out that for linear systems, if you do this again for linear systems, you can show that mutual information between x and y can be written in terms of log of 1 minus the coherence, which is a very nice simple equation, quantifying again coherence, but based on mutual information. The point is that mutual information is very universal. This is just for linear systems. Therefore, this could give us a way to define this thing for nonlinear systems. And that's what we have done. And that's what has been done before, actually. And the game plan is very simple. These are our time series. All right? And if I want to find out the coherence between this signal and that signal, all I need to do is take those two signals, take them to the Fourier domain, compute the Fourier transforms, compute the joint densities of the new data time series, the new data, and then compute all the other quantities that I want, and eventually compute the mutual information between them in the frequency domain. All right? And that would give us a concept of coherence between the two signals. Then you may ask, does it work? You can do that at different frequencies as well, because these are just two data sets that have different frequencies, and you can do any pairs of frequencies you want. The question is, does it work? And the answer is obviously yes, otherwise I wouldn't talk about it, right? I would skip that part. It works. It works beautifully, actually. And if you go back to this example that we have talked about before, x is just a simple cosine, y just that cosine square, right? And if the frequency f was 4 Hz, what do you think would be the coherence between x and y? All right? Y is just x squared. Therefore, x squared is a cosine squared, right? Therefore, it would be cosine at 0, cosine at 8 Hz, right? Therefore, you imagine that you get something like this. And that's exactly what you get. This is a heat map of coherence between x and y. There is coherence between them at 4 Hz, and 0, and 4 Hz and 8 Hz, right? Exactly what we suspect. And you could get all of that from actually doing this thing for a long time, creating x and y, compute the joint densities and all that. You can get this plot relatively easily, right? And that gives us some hope that we can indeed find out the coherence between two signals that are not linearly related to each other, and it makes a lot of sense. All right? Going back, in summary, that's where we are right now. We just want to take our data in the frequency domain and analyze the coherence at different frequencies. Now, the second thing that we want to talk about today is influence, is causality. All right? Therefore, we finished with coherence. We gave some examples of that, and it does work. The next metric that's going to give us another type of graph would be the one that is going to try to measure causal relationship between the time series that we are recording from the brain. All right? Now, I'm going to speed up a little bit so that I will be able to show the results and not be cut off. Causal relationship is what we're interested in, and this is also an old problem that people have been looking at it for a long time. The list of luminaries that have been working in this problem is not that long, but they're very sort of well-known engineers and economists that have been working on this thing for a while. and it is a concept of causal relationship. And the one that we are focusing on is the one that is universal and can be used to quantify causal relationship between time series that are not necessarily related linearly with each other, right? And again, it requires you to not just use mutual information in its raw form, but then adjust the mutual information so that you could bring in the concept of time and causality into this picture. It's called directed information. It's been around for a while, and it is somehow analogous to the concept of mutual information with the subtle differences here. And it is very well defined. You can compute it for any time series that you want and sort of look at the time lags between them and then what causes what. All right? And it can be computed because it is very universal. As long as you can compute entropies of the time series, you can compute mutual directed information, just like the coherence that I mentioned earlier. And again, I don't want to go through this example in detail because I don't want to run out of time. Basically, what it does is that you can invoke chain rule and then write this down as a number of different entities and compute them and see what you get. And but then let me give you a couple of examples to convince you that it's an intuitive idea. All right? One example is this, that very famous example of a channel, let's say, that you have x as your random variable that you're interested in, times that you're interested in, and y is the one that you observe, and z is just annoying noise, right? The question is that if these are the assumption and x and z are independent from each other, what is the directed information between x and y, right? Does x cause y? Yeah, right? Because if you know x, it could help you estimate y, right? Then the other question is that does y cause x? Yeah, because the indices are the same. My current y can help me understand the current x. Makes sense, right? And if this is what we're going to get, therefore this is a useful quantity. If it's not what we're going to get, then there's something wrong, right? It turns out that's exactly what we're going to get. If you break this down, as we did before, like this, then you could follow these terms and recognize that at the end of the day, it is this quantity here. And some of the terms are 0, some of them are non-zero. It all that depends on the indices x and y that they have. Are they helping you with understanding of the other, right? If you do the same thing and try to compute directed information between y and x, does y help you understand x? Because of the fact that the indices are identical, yes, the answer is yes again, right? Now, what if we change this to this problem? What if we goof around with the indices a little and get this? Now, in this question would be this. In this case, it would be the same situation as before. X and y, z are independent because noise under your signal. The question is that does x help you predict y? Yes. Does y help you predict x? No. And if you do the math exactly the way we did before, that's exactly what you're going to get. That directed information between x and y is what it was before, and directed information between y and x is just flat out 0, right? Because just the way the indices work out. Basically, the point is that this formulation helps you, sort of focus on the indices and the time samples and all that. Right? If all of this set is done, the question, and then if you believe in the fact that directed information could help you quantify causality and help us understand our graphs, this is what we're going to get. We can compute that because we have data, and then from the data we can get the entropies and the conditional entropies and all that. And then this is going to be our pipeline again, that we have the data from this experiment that we just did, and these are just different electrodes, and we repeat the experiment 500 times. We can compute the joint density, we can compute the entropy, and then compute the directed information between these two time series, or any pair of time series that you want. All right? Again, that's the kind of thing we're going to get, right? I was just explaining it in depth. Right? This is what we're going to get. We're going to get graphs like this. And we're going to have two sets of graphs to see what we can learn from these two sets of graphs. One set would be directional graphs. One set would be undirected graphs, trying to tell you different things about the experiment, right? And at the end of the day, we are very interested in dynamics of these graphs, right? All right. Now, how do these, let's first focus on undirected graph. Therefore, we have these nodes, these vertices that are basically our time series. And then we draw edges between them that are basically coupling at high gamma. At high gamma, coupling between these two vertices, right? And that's what this is. And I'm looking at the dynamics of this undirected graph as we go after stimulus. The stimulus is presented to the patient. This is how the connectivity is for that particular patient. At articulation, that means the patient has seen the image and is beginning to articulate the word alligator. This is how the coherence is. And after articulation, this is how the coherence is. And the question is, what are you going to do with this? Right? That's a very fair question. And we are sort of analyzing all of that and trying to sort of come up with better and better graphical models to model the data that we have. All right? But it's just one set of results that we have. All right? Now, there are other things you can do with these kind of graphs because you have all these nodes and all these vertices and all that. There are tons of metrics, graph theoretical metrics out there that you could see what are interesting to your problem, right? One thing that you could do easily is to look at your graph and then say, what is the density of the graph, right? What is the density of the graph, right? You can define it very formally to be something like this. The D being the degree of a particular vertex, particular node, and then compute the degrees divided by what is the potential number of degrees you could have, and that would be the density of the graph, right? And it would be good to look at the density of the graph to see how it changes across your experiment. Right? For undirected graph, this is how it looks like, all right? Kind of interesting, a little weird. Basically, right after stimulus, there is a sort of a large density in the graph, and then it goes to a dip, and then after articulation, the density increases again. Right? Just a way to see the data, a way to infer from the data. Yes?
So this is averaged over time or patient?
This is for a given patient. We only have 7 patients. For a given patient, and this is windowed data because we have many trials windowed over the same period at different trials. And this is different time periods during the experiment. Therefore, this would be the time of stimulus, 0. 20, roughly 20, is the time of articulation. And this is the time behind articulation.
So you said there are 500 experiments for each patient.
There are 500 trials for each patient, yes.
The average over the 500?
Yes. Yes.
And what's the variance? Is it too large?
We did all sorts of statistical tests to see the significance of the results, meaning null hypothesis testing and p-test, p-scores and all that. And then we came to the conclusion that these are statistically significant results. And you realize the limitation. The limitation is that that's all the data you have, right? Therefore, all you can do is that see if that's enough for that statistics that we're measuring. And we did all of that, right? Okay, and again, we have a few things to work with. One of them is that 500 time trials that we have. All right, now, then if you focus on undirected graph for a second, see what we can learn from undirected graph. No, undirected graph we already did. If you want to focus on directed graph, we want to see what we can learn from directed graph. All right. For directed graph, we have done a few different analyses and at different scales, which is kind of been fun. One is at a very coarse scale, like looking at the density of the whole graph, basically. Another one would be to look at the clusters and basically have an intermediate scale view of the graph. And the last one is to have a finer scale analysis, coarser, finer scale analysis of the graph to find out in degree, out degree, and then look at graph points specifically. Let me quickly run through these things because I have only like 8 minutes left. All right? Now, at the highest coarsest scale, let's just focus on graph's density. Once again, this is what you get. Again, averaged over trials and all that. Same story as before, just at the different graph because the metric is different. Right? As you see here, right around before articulation, there is an increase in density of your graph, density of the directed graph. Right? Basically, the patient is thinking, is trying to articulate, and then this is what you're going to get. This is we will see that across the space in a few minutes. Right? This is the density of the whole graph. Right? That's for one patient, and this is for another patient. Patient to patients are different, but still you see the same increases that we saw before in the other patient. Definitely patient to patient are different. Right? Now, but we still see an increase in density right around articulation. And the second level question that we had was about clusters. Are we seeing some obvious cluster in our graph? Right? Whenever you think about clusters, you think about something like this. And then you realize that defining a cluster is not easy. Right? What is a cluster? A cluster could be one node, one vertex cluster. You can say, oh, everything's a cluster, right? That definition is not trivial. I mean, you can define something and then see if you're happy with that. This area of clustering and modularity has been studied in graph literature quite a bit. And then one that we have focused on is called Louvain clustering, very standard. And basically it tries to give you a graph with all the vertices and edges and tries to define clusters in a meaningful way. And not only defines it in a meaningful way, but it also gives you a protocol to actually estimate it. Right? And it's quite practical. The protocol is quite practical. It basically looks at the density of a whole bunch of nodes and connection between these sets of nodes, and then comes up with a very sort of intuitive way of saying, oh, this is a cluster, that's a different cluster, and all that. Right? And that's what we exercise in this case. And one thing you notice is that this is the number of clusters that we found based on the data. And this is the density of each cluster that we found in the data. As you could notice here that right before articulation, the number of clusters increased. And and then the number of connections per cluster increased right before articulation, but then it dipped down for a while. Right? And then that would be one view of that problem. Another view would be this, that if you look at this, and this would be a stimulus onset, and this being the articulation time, these are different clusters that we found using Louvain's protocol, and they're being colored differently, and the same brain, and then you realize that as we go through these time periods, you see that the number of clusters are roughly staying the same, but the density of the clusters are changing quite a bit, right? And this is some observation that we made that is kind of interesting to look at, right? And then another layer of analysis would be to look at the finer scale of this particular analysis. Again, to look at individual electrodes, to look at their in degree and their out degree, to see how are they impacting other electrodes, other nodes, and then look at the dynamics of it across time. And again, this is articulation time. This is the onset of the stimulus. And you see that these are all very sort of useful ways of depicting the data that can help us understand the dynamics better. And then again, you could sort of step back and then look at the density of these kind of in degrees as time goes on. And you recognize that, again, the density increases. as articulation happens. And overall, if I want to step back and leave some time for questioning, what we have done is that we have the data. We have the data for this particular language production. And all we have done is try to sort of use some data analytic tools to sort of package the data in ways that we could make statements about them. One of the things that we found out was that there is increased functional and effective connectivity at the onset of a stimulus and at the articulation time. And that during the articulation time, the cluster seems to be much heavier than times before. And the question is that how can we sort of move on from this set of analyses and make statements that are helping us look underneath what has been known already. All of the results that I'm showing you, they do not contradict the statement that I mentioned at the beginning about the flow of information from visual cortex to other regions and to Broca region. They do not contradict any of that. They just they tell you a little bit deeper analysis of the actual network and how it changes across. Right? We were not planning to contradict anybody's results because those have been established results for 30, 40 years. It's just that we just wanted to understand it a little bit at a deeper level. And we did that with this analysis. All right? I am done, and I'm happy to take questions. It's been a very quiet audience today. Questions? Yes, sir.
I wonder about the time series. You say the brain has 86 billion neurons, and you measure spatially at 200 some points. How do you know that you're not spatially undersampling the processes that go on?
The question is that how do we know that we are not spatially undersampling the data? Possibly we are. I mean, what people do is that whenever you have data like this, you worry about sampling of the Nyquist rate type, right? And then you also worry about spatial sampling. These are all typical challenges that we all face. We know that the data that we have is raw. Therefore, our sampling of a kilohertz is pretty standard because it's been studied that these kind of signals, because of the neurophysiological properties of our brain, we know that they don't go faster than that. That's fine. But the question of spatial sort of sampling is definitely there. There are limitations, right? There are limitations to the actual electrodes. Let's say ECOG electrodes of 10 years ago, they only had like 50 electrodes. Now they have improved the density of them. Now they have up to 250 to 300 electrodes. Therefore, the region is still similar, that the electrodes are much closer to each other. Therefore, the spatial sampling is improved. At the end of the day, we are at the mercy of the experiment, mercy of the product that is out there today. And so far, that's all we have, right? Now, if our colleagues, neurosurgeons, give us a better set of electrodes with finer resolution, that would be great. I doubt that would make that much difference. Ideally, you would like to probe every single neuron, right? But that's never going to happen. Therefore, from individual neurons to grids that are like 4 millimeters apart from each other is a long distance between them. And that's where we are today. And we're never going to get to individual neurons. And I'm not even convinced that if we make the grid denser, it's going to give us that much more information. Because again, these recordings are from the surface. And then some of the regions are a few centimeters deeper. Therefore, by the time the signal reaches the surface, has gone through so much already filtering. Therefore, I think some of the resolutions are definitely lost. That's my best answer. Any other question? Yes?
About the clustering, I wonder if how can we relate the measure of clustering in your analysis? What is it? Like, can it be related to sort of synchrony or having this similar frequency in each region, like the neurons that fire together, wire together, or something like that?
The analysis that I showed earlier, a few seconds ago, were for directed graphs. Therefore, it was not for coherency. But we could do the same clustering for coherency graphs, right? What I showed you was for directed graph, the influence. There we thought that for now, we are looking at influences and causality, and we want to know each clusters of these causality picture. Therefore, see there's a cluster of data here, and then influences next one, all that. And that's what we did it like that. But I could easily do similar kind of clustering for coherence at high gamma or other frequencies. And this is kind of the things that we are doing now to sort of complete the picture. So far, and this particular paper that I showed you earlier is all about directed information. It's all about the causal influences and all that. And, but we do have the results for coherency, but we haven't sort of completed it yet to be in a publishable form yet. But the data is there, the analysis is there, and then we will look at that. Right now, we're just trying to understand. We're trying to understand and we're trying to sort of validate the whole Broca region analysis and try to understand specifically at the graph level, what does that mean for all that? And that's why we focus on causality for now. Yes, sir?
So, so you have pictured these graphs evolving during the time.
Mm-hmm.
So is this that you fixed a distribution for the whole signals and then evaluate the distributed information for this whole, or did you assume like windows that so if you assume the whole process of speaking, so there is one part that you rest and then the one part that you try to speak. So did you assume different distributions for this different part, or you assume the same distribution and then try to estimate this for the?
Now, should I repeat the question or have we finished recording?
No, repeat the question.
Okay. The question is that how did we do these things, and did we fix the time? Did we fix the clusters, and then did we look at sort of estimated the densities and all that? You gotta, what we did was this, that if this is the recordings, and if this is sort of, let's say, a graph of n densities, and then across time, the time is going from A to E, right? And in that time period, which is a long time period, we have periods of data that we recorded, and then those are time windows for estimating the density of X, density of Y, and all that. For a time window, we did that, and then we slide the time window across A to E, right? For each time window, we computed the graph and computed the clustered the graph for each time window at around A, around B, around C, around D, and around E, right? And then we watch these graphs for those time windows around A and B and all that, watch their progression in time. Now, did I answer your question? All right. Yes.